Isn't it just amazing when you stop and think about, well, us, humans, what really makes our brains tick?
It really is. I mean, the sheer complexity.
Right. We compose music, we question reality itself. And lately, there's been some really almost sci-fi level research getting closer to the why.
You're talking about that study, the one the debrief reported on.
Exactly. Scientists putting human DNA into mice.
Hmm.
And get this, their brains actually got bigger, significantly bigger.
Yeah, and it wasn't just random DNA, was it? They targeted these human-accelerated regions. Dollars.
ARs, right. Tell us a bit more about those.
Well, basically, they're parts of our genetic code that change really fast, evolutionarily speaking, compared to, say, chimps. And the idea, the strong suspicion, is that these speedy changes hold major clues about what makes us uniquely human, especially brain-wise.
Okay, fascinating stuff. Welcome, everyone, to Dansplain. This is where we dive deep into topics just like this one. I'm Edward.
And I'm Lily, and today, we're unpacking this incredible study and sort of the whole scientific picture around it.
Our mission, if you will, is to really get under the hood of this experiment. What did they do? What did they find?
And how does it connect to other research? What's it all telling us about, well, the evolution of our own intelligence?
So if you're ready for some truly mind-bending science, you're in the right place.
Absolutely. And hey, if you like what we do here, please do hit like, subscribe, you know the drill.
And check out www.dansplain.xyz. We've got more deep dives, articles, all sorts of stuff over there.
Okay, let's jump right in. The core experiment. These human-accelerated regions, or HER, and specifically one called HER5. Why HER5?
Right, so HER5, it's interesting because it's not a gene that makes a protein directly. It's more like a control switch.
For a regulator, yeah.
Exactly. It dials other genes up or down. And the crucial thing is the human version of HER5 looks quite different from the version in other primates.
Very different, which strongly suggests it was, pretty important when our evolutionary paths split.
So the experiment was basically take the human HER5, put it into mice.
That's the core of it, yeah. They engineered mice to carry the human version instead of their own.
And the result?
Quite remarkable, actually. These mice, on average, had brains about 6.5% larger than the control mice.
6.5%. Now, that might not sound huge to everyone, but in brain development terms.
Oh, it's significant. Even small percentage changes in brain size can mean big differences in structure and potentially function down the line.
And the researchers think they know why this happened, right? Something about radioglia cells.
Yes, exactly. They found more radioglia in the modified mice.
Okay, refresh my memory. Radioglia are?
They're super important neural stem cells. They do two key things during development. First, they divide to make more neurons, literally the brain's building blocks.
Got it. More neurons.
And second, they act like a scaffold, kind of guiding these new neurons to the right spots as the brain grows, especially that outer layer.
That outer layer. You mean like the cortex, the thinking part?
Precisely. The expansion of that outer layer, the neocortex in humans, is massively important for our higher cognitive functions, reasoning, language, all that stuff. So more radioglia could mean a bigger, better organized cortex.
Wow. Okay. And they pinpointed why the human HER5 did this?
They did. They found 4 specific genetic tweets, mutations, within the human HER5 sequence.
Just 4?
Just 4 tiny changes that chimpanzees, our closest relatives, don't have. And the evidence points to these 4 changes being the reason HER5 boosts radioglia production.
4 little letters in our DNA making such a difference? It's kind of mind-blowing.
It really is.
But okay, the big question. The elephant in the lab, maybe. Did the mice get smarter? Did they start solving tiny mazes faster or, I don't know, planning tiny mouse coups?
Yeah, that's what everyone wants to know. But cognitive testing wasn't really part of this specific study.
Okay.
So we know their brains were structurally different, bigger in a specific way. No. But whether that translated to higher intelligence in these mice, we don't actually know yet.
So that's definitely a watch this space kind of situation.
Absolutely. A critical next step for whoever follows up on this research.
Right. But like you said earlier, this mouse study isn't happening in a vacuum. There's related work going on. You mentioned organoids, brain organoids.
Yeah, it's another fascinating angle. Brain organoids are like, well, miniature simplified brain structures grown in a petri dish from stem cells.
Mini brains in a dish. Wow.
Kind of, yeah. They mimic early brain development. And researchers use them to compare human HER5 directly against chimpanzee HER5.
Did they see the same effect as in the mice?
They did. The human HER5, even in that dish setting, led to more radial glioma and just generally more growth compared to the chimp version.
Okay, so that really strengthens the case for HER5 being a key player in human brain expansion.
It provides really nice converging evidence, exactly, without needing to, genetically modify primates.
Which is obviously a huge ethical consideration. Speaking of related genetics, what about that Neanderthal DNA research? That sounded intriguing.
Yes. So, other groups have been putting Neanderthal DNA segments into human brain organoids.
Seriously, what happens?
Well, they've observed some subtle differences. The shape of the organoids can be slightly different, and the way the neurons behave, how they connect, might differ too.
Subtle, but maybe important.
Potentially very important. Neanderthals were smart, no doubt, but maybe these small differences in brain wiring or development gave Homo sapiens an edge somehow. It's speculative, but suggests differences in how our brains were fundamentally organized.
It's like digging into the evolutionary code of different brain operating systems.
That's a good way to put it. And it's not just about modifying existing stuff like HR5, there's also research on totally new genes.
New genes, like appearing out of nowhere.
Sort of, yeah. They're called de novo genes. A study in Nature Ecology and Evolution found about 74 genes that seem unique to humans, no obvious ancestor gene in other primates.
And what do these new genes do?
Well, here's the kicker. When scientists put some of these genes into mice, they also saw effects. In some cases, bigger brains again, and interestingly, some apparent improvements in memory tasks.
Okay, so it's not just one magic bullet like HER5. It's HER5 changing regulation and completely new genetic tools appearing.
Exactly. It's likely a combination, and we shouldn't forget about gene duplication either.
Ah, yeah, the old copy-paste errors in DNA.
Right. These duplications create extra copies of genes. The original keeps doing its job, but the copy is free to mutate and maybe gain a new function over evolutionary time.
So that creates raw material for evolution to work with.
Precisely. It's a major engine for creating complexity, especially in nervous systems. So these specific changes in HER5, or the de novo genes, are happening against this broader backdrop of gene duplication events that allowed our genomes to become more complex in the first place.
It paints a picture of, well, layers upon layers of genetic change building up over millions of years.
It does. From accelerated regions like HER5 to organoid comparisons, Neanderthal insights, new genes popping up, ancient duplications. It all points to a really intricate genetic story behind our brains.
Which naturally leads to thinking about the bigger picture. What are the implications of figuring all this out? You mentioned medicine.
Absolutely. Understanding the normal blueprint for brain development, the genes involved, the timing, that could give us huge insights into neurodevelopmental conditions. Things like autism spectrum disorder or schizophrenia, where something seems to go differently during early brain formation.
If we know how it's supposed to work, maybe we can figure out how to help when it doesn't. Makes sense.
That's the hope, yes.
And you also mentioned AI. How does AI-5 relate to artificial intelligence?
Well, it's more indirect, but AI researchers often look to the brain for inspiration, right? For efficient designs, for ways to learn and adapt.
True. Biomimicry.
Exactly. So understanding the biological algorithms or principles that guide brain development, maybe even the specific pathways influenced by things like HER5, could potentially offer new ideas for building more powerful or even more brain-like AI. Learning from nature's R&D.
Learning from the master architect.
Yeah.
But it's kind of daunting. Well, you said HER5 is just one of about 3,000 ACAR.
Yeah. 3,000 known human accelerated regions. We've really only focused on a handful so far.
So we barely understand how this one piece works, let alone how all 3,000 might interact.
That's right. How they all work together, synergistically perhaps, to triple our brain size compared to our earlier ancestors. That's still a massive unknown, a huge area for future work.
Because future directions would definitely include, well, testing those HER5 mice for cognitive changes, finally answering that question.
Definitely. And also trying to understand the network effects. How does HER5 interact with other HER5s? Or with those de novo genes? It's likely a very complex web.
Which brings us, inevitably, to the ethical side of things. I mean, putting human DNA, even regulatory bits, into animals. That raises some questions, doesn't it?
It absolutely does, and it's crucial we talk about it. There are immediate concerns about animal welfare in these experiments, of course.
Right, but also bigger, maybe more philosophical worries.
Yes. Concerns about blurring the lines between species. If we start giving animals genes strongly associated with human brain development, what are the potential outcomes? Could we inadvertently create animals with more human-like cognitive abilities or awareness?
That sounds like the plot of a cautionary sci-fi movie.
It does touch on those themes. And then there are the unknown risks. What if modified animals, say these mice with enhanced brains, were to escape into the wild? What ecological impact could that have? We just don't know.
Have you seen much public reaction to this kind of research? What are people saying?
Oh yeah, you see it on platforms like X, formerly Twitter, and elsewhere. It's a real mix. Some people are absolutely fascinated, amazing clues to human evolution. This could cure brain disease.
The potential upside.
Exactly. But then you also see a lot of concern. Comments like, should we really be playing God? Or are we going to end up with mice writing symphonies? Maybe slightly jokingly, but the underlying worry is there.
Sci-fi plot gone wrong, someone probably tweeted.
I'm sure they did. And it's important to acknowledge those anxieties. This research pushes boundaries, and it's natural for people to question the implications and whether we have the wisdom to handle this kind of power responsibly.
So it's not just about can we do it, but should we do it, and if so, how?
Precisely. The science is incredibly exciting and holds immense promise, but it has to proceed hand in hand with open discussion about the ethical lines we're drawing and the potential consequences, intended or otherwise. Public discourse is vital.
Okay, so let's try and wrap this up a bit. Key takeaways. This HAR5 study is a really big step, wouldn't you say?
I'd say so. It's a concrete example of how a specific piece of human unique DNA can directly influence brain development in a way that mirrors our own evolutionary path, at least in terms of size increase via radial glia.
And it connects with all this other research. The organoids, the Neanderthal DNA, the new genes building a much richer, albeit still incomplete, picture of how our brains became, well, our brains.
Right. And the potential implications are huge. For medicine, maybe even for AI down the road,
But there are those significant ethical questions that need careful consideration as we go forward.
Exactly. It's a powerful reminder that Haiti 5 is like one important instrument, maybe a genetic maestro, in this huge evolutionary orchestra that built the human brain. We're just starting to identify the players and figure out the score.
So a final thought for you listening. As we keep pulling back the curtain on our own origins, on the very biology that makes us human, how do we balance that quest for knowledge with our responsibility?
Yeah, where do we draw the lines? What does this research fundamentally tell us about ourselves? And what obligations does that knowledge bring? Something to definitely mull over.
A lot to think about indeed. Well, that's all the time we have for this deep dive. We really hope you found this journey into brain evolution as fascinating as we did.
If you did, please do give us a like, subscribe for more, and definitely head over to www.dansplain.xyz.
We've got loads more content there, episodes, articles, you name it. Thanks so much for tuning in.
Thanks, everyone.